is a type of cancer that occurs in the cells of the cervix. Now, this cervix is actually it is the lower part of the uterus. Mm, and in a long-lasting infection with certain types of human papilloma virus is the main cause of cervical cancer. So, this human papilloma virus, long-lasting infection with certain types means genotypes of human papilloma virus is main cause of cervical cancer. There are a lot more genotypes of HPV human papilloma virus. Even normally we carry so many human papilloma virus on our skin, but there are certain types. If they actually are responsible for long-lasting infection, then actually they cause this cancer, cervical cancer. And of course, this cervix is only present in females, so this cancer occurs in women only. Now, uh, if you look at the anatomy, this is the uterus. We know that this is ovary, then this is uterus, and this lower part of the uterus, this is called cervix. So the cells go wrong at this side. Okay. Now again, this is the lower part of the cervix where this occurs. Therefore, it is called cervical cancer. And this is very much treatable cancer. And of course, hygiene is very important. Uh, so mm, and now vaccine is also there. Though it is not as popular as polio vaccine, but there are vaccines for HPV. Uh, so, and very much treatable, and early detection could be made. You see, this is the mm, end of the lower end of the cervix, where you can find these white patches. These actually the cells are actually going abnormal, and depending on the abnormality, the extent of abnormality, you can classify the cells. Okay, some are precancerous lesions, and some could be actually cancers. Now, what people do, they collect sample from this site and examine under microscope to assess the extent of or the to what extent the cell has gone abnormal. Now, this cervical screening test is very much popular for early detection of this cancer. And this is the algorithm people actually use to detect the HPV. They collect the sample and try to detect the HPV, because in most of the cases, as I said, HPV is the main cause of cervical cancer. For whenever they find the abnormality, they collect the cell and they first try to go for HPV test. And if it is positive, then they go for another test. We'll explain this. And if it is again positive, then they can go for the therapy. Okay, so and this therapy is really very effective, and they can remove the abnormal tissues, and the patient can be recovered nicely. And if test is negative, and so people are asked to repeat the test after 5 or 10 years, or as required. Then again, if HPV is positive, but in this VIA test it is negative, then they again go for this repeat test, uh, uh, but no need to go for this therapy. Okay. Now, if we look at the epidemiology of this is now cancer, cervical cancer in women is on the declining trend in India. Uh, so, but but still, the substantial amount of uh, a number of Indians women actually get affected with this disease, this decline. Now, India accounts for one-fourth of cervical cases and deaths worldwide. So, if you consider the whole cases worldwide, then India actually contributes one-fourth of them. Then you can understand the gravity of the disease in India, and it is still there is high burden of cervical cancer in developing countries like India. Is largely due to the lack of screening that allows detection of the precancerous and early stages of cervical cancer. So screening is very important, and then of course hygiene. Uh, so uh, this is how if there are some lessons, if we go for this VIA, the visual inspection with acetic acid. This is VIA means visual inspection of acetic acid. So people apply certain 5% acetic acid on the mouth of this cervix, and then this lessons abnormal cells appear in different way. So so on looking at the type of the this I mean this opaque cells, uh, uh, we can grade these lessons, whether it is LSL, LSIL or HSIL. Uh, LSL is not that much mm, uh, dangerous. Even HSIL also not that much dangerous. They can always revert back to normal. They're not cancer. They're pre-cancerous lesions actually. Okay. From LSL it converts to HSL. Then from HSL it moves slowly to the cancerous stage. So just to have an idea what is the stage of this lesson, you can simply put 5% acetic acid and look at the cells, how they look. On the basis of these characters, you can, yeah, actually it can be diagnosed. Uh, so what happens actually, acid coagulates proteins of nucleus and cytoplasm and thus make the cells opaque. So that's why you can see the opaque cells, and the dull white plaque or with faint border. There you cannot define the border of these white opaque plaques. Then it is most probably LSL. And if there it is if the thick plaque, there is thick plaque with sharp border, probably this is the HSI. Now, if we look at the histology, then this the normal histology, it looks like this. Or if we collect some portion, small amount of tissue and go for histological slides, then it looks nice. This kind of appearance. And when LSL is there, then you can see the changes in the histology. Okay, there is proliferation of cells. There is some changes in the morphology, and in case of it is HSL, then even there is sharp changes in the morphology of the cells, appearance of the cell. The nucleus is different, and if it is then uh, goes to this cancer stage, then actually you see they have started invading. Okay, these are the stages from normal to LSL. These are pre-cancerous; they are not cancerous. Okay, so that can be easily detected. Now, This HSL means high-grade squamous intraepithelial lesions. They look somewhat to very abnormal when looked at under microscope, and they are usually caused by chronic infection with certain types of human papillomavirus, and are found when a PEP test or biopsy is done. Now, what is PEP test? PEP test and VIA is different. So this is actually VIA. When you put the acetic acid, then you see the outer appearance of the cells on the mouth of the cervix. But pap smear is it is, you need to collect the sample from there, and then you spread it on the slide, and you go for the uh, histological screening. Now this is how actually uh, it looks under microscope, or specifically actually it looks like something like this. Yes, you can see this kind of changes. And I have slides because our work is going on in our lab on this. So you can see this epithelial cells, normal epithelial cells, exactly like this. And if the changes, you can see the uh, okay. So then this is what actually this is how they look under microscope. You can see this kind of cluster. You can see dividing cells, more dividing cells, and on the basis of this, gradation could be made nicely. Uh, and another is histology. So if you go for histological then slide, you can see this kind of histological sections. In histological section, you can see this kind of changes. Okay. Now the term of HSIL or LSIL, they are actually replaced with CIN1, CIN2, CIN3. Okay, LSIL is called CIN. It is actually mild dysplasia. Yeah, and HSIL is CIN2 is moderate dysplasia, and CIN3 is severe dysplasia, and it also the also carcinoma in situ is also comes under CIN3. So now and microinvasion, yes, invasion is still there, but invasion is not there in CIN3, so it is still under HSI. So correlating cytology and histologic terminologies for neoplastic squamous or epithelial changes in the cervix. So you can find this term CIN somewhere in some literature, CIN2, CIN3, or in some literature you will find LSIL, HSIL. So now you can understand uh, what is the changes in these cells. This is the normal non-dysplastic epithelium. So we also discussed all this dysplasia and hyperplasia in earlier episodes. Now, definitive, what happens if you find this? What could be the treatment? So, definitive treatments are there depending on the age, uh, desire of reproduction, risk factor present, degree of dysplasia, and facilities available of popular. So, depending on this, people may go for different kind of treatment options. There are local ablation with cryotherapy, cold coagulation, electrotherapy, and laser preferred. So this is local uh, ablation, and excisional is bone excisions and large lobe excision. So and overall hysterectomy of the whole uterus can be removed. So depending on the all these things, so clinician decides what would be the best one for the uh, patient. Uh, Also depends on the uh, degree of dysplasia is very important. Okay, so maybe some local ablation is fine. Uh, then screening, as I said, screening is usually done with the pap smear. So some sample is collected through spatula or side to brush, and this is taken in slide, and then stained. It is stained and looked under microscope. Now for HPV, in the same samples, some samples is separated out for HPV detection. So samples are HPV DNA is detected in the other part of the other half of the sample. So polymerase chain reaction could be used and hybrid to capture this. So polymerase chain people do mostly polymerase chain reaction to detect the HPV DNA. Now, again. So there are some clinical symptoms of the this is uh, well, right, this is what the pap smear is. So the sample is collected uh, and then this collecting cells that is collected from cervix, from the lower narrow end of the uterus, and they are detected uh, after staining, and the changes. Pap smear can also detect the changes in the cervical cells that suggest cancer may develop in the future. So, looking at the changes in pap smear, we can say what stage the patient is at what stage. Uh, so the patient um, is, what the disease is, and what could be the proper treatment for that. Now, so it's because early detection is very important, and so it can be prevented nicely. These are the diseases. Well, now mm, the screening modalities we discuss about VIA, visual inspection of cervix with 5% efficacy, and this is 50 to 80% sensitivity of the test is there, and it is very low cost uh, and ease of people can be easily trained to test this, and results are immediately available, amenable to screen and treat approach. Now, subjective test and false positivity is high. This is what. So those people who are looking at it, depending on his or her skill, uh, some false positivity could be there. So, but overall, the VIA is very low cost, rather clinical observation, by rather screening process, uh, by which one can be easily screened even in remote areas. Uh, and then comes pap smear. So VIA is one of them. Then pap smear is second. You have to collect the sample from that area and stain the slide and look under the microscope. It is more specific. Pap smear is more specific screening test. And then third is isolation of DNA and to test for the presence of HPV, human papillomavirus. These three things are screening tests are widely used. And then prevention. Uh, for prevention, it is if the girls between 9 to 14 years. So HPV vaccination is very important because you see in this is graph, this is uh, is very important, very important. If you look at the incidence of the disease age-wise, you see before 15 years there is almost nil. Incidence is very nil. Okay, so HPV infection is age specific, right? Then after this at adolescent stage, infection starts rises sharply, and you can see that as the age progresses, so then infection actually uh, going down. Uh, so if you want to vaccinate the population, then what would be the favorable age? Can you suggest? Hello.
So before 15 years.
Before 15, of course. So if infection is there, then it is gone. I mean, what is the use? But people use, there are certain modalities, but it is always, recommended that girls should be vaccinated at least before 15 years of age. Then they can raise the antibodies, uh, and so they can resist the infection of HPV. And if HPV infection is not there, there is, you can say, no chance of getting this disease because most 90% cases HPV is responsible for the disease to occur, right? So if HPV is not there, then there is very little chance to get the disease. Okay, now this is what now HPV in cervical cancer, as we are talking about. Now cervical cancer remains one of the gravest threats to women's lives worldwide. Okay, so cervical cancer, so it's it's a gravest threat, and of course worldwide. Now, cervical cancer is caused by high risk type of HPV. So human papilloma virus. So I told you that there are several kinds of HPV, types of HPV genotypes, several number different mm, mm, types of HPV genotype is available in the environment, and there are certain types. They're innocuous, they're innocent, they don't do anything to us. Even normally we are having on the in our body, but some genotypes like HPV 16 and HPV 18, they're the most common high risk HPV types for in cervical cancer. There are some more high risk genotypes, but they're the most commonest genotypes that are responsible for cervical cancer. Now, they're responsible for approximately 70%. I would say they are responsible for 90% of cervical cancers in some places. Uh, then HPV is currently the most common sexually transmitted infection. Also, this is a STI, common STI. Then 80% women can be infected at some point in their lifetime, and most infection is clear naturally. Yeah. One thing is that this infection is self-limiting. If people get infected with this, even without vaccination, uh, they get cured naturally. self-limited. Okay. They do not survive this HPV 16, 18. They do not survive in most of the individuals, but some cases they persist, and we do not know that in which person or in which girl or woman they will persist, and therefore vaccination program is very good. If somebody is vaccinated, then uh, is mm, rather that uh, she is uh, more in a side of not getting the infection. Uh, then cervical cancer has long preclinical phase from persistent HPV infection to development of cancer. This is where the importance of screening lies, because it's a long there is long preclinical phase. So even if it is there, uh, and if it is detected in the screening process, then immediately this action can be taken, remedial treatment can be taken, and that can be cured. This is another advantage of this is because they have long preclinical phase, which is not there in pancreatic cancer. uh, it's very fast developing cancer, the pancreatic cancer. Now cervical cancer uh, uh, actually primarily caused by HPV we have talked about. Now what is HPV? HPV is DNA virus and has over 100 documented genotypes. So I talked about this. There are a lot of uh, variation in HPV genotypes, and there are 100 documented genotypes. And out of this, 40 are known to infect the anogenital tract, and between 12 and 14 are high risk genotypes, those that can cause progression to cancer. Now, cervical cancer is slow growing. Its progression through precancerous changes provide opportunities for prevention, early detection, and treatment. This is very important. And cervical cancer can be altogether eliminated. This can be altogether eliminated. So, another thing is that I always tell that hygiene is very important. So don't get people shouldn't get infected. So if proper hygiene is there, there is no chance to get infected with the virus. Okay, then we have come to the end. So HPV is the major cause of the cervix, cervical cancer. Pap smear among the other tests, so is the best for early detection, and presentation may be asymptomatic, especially in the early stages. Treatment depends on the stage of the disease, and survival can be very high when treated early. Okay, so with this, we've come to end. So any question? So it is clear, right? So in the next class, we will move a bit deep into the mechanism how this HPV or the oncogenes in HPV actually cause the transformation of the normal cell into cancer cell. Okay. So we stop.
Okay, sir.
Okay, sir.
Okay. Later we'll I'll share all these slides with you. Okay.